Hello, this is Nanadazigansa, and I want to do a little short talk about mRNA vaccines. Okay, and we've all heard the information or the news about uh, the developments being made by Pfizer and BioNTech as well as Moderna with their mRNA vaccines against COVID. And to help people understand what's going on and dispel a lot of the disinformation out there, this is just a very short um, explanation with my really bad drawing to explain what this is. Also, let's get started. Um, Classically, to make a vaccine against any pathogen, you would take the virus. So I'm going to draw a virus here. Don't laugh at me. This is my virus. We'll call this virus X. This is the envelope of this virus, the envelope, and it has genetic material in here. So this is an RNA virus, so that's the RNA. And what we'll do is we will try to kill virus X, either by heating it a little bit or applying certain chemicals that um, kill the virus X, not all the way, but a little bit. And we call this attenuation. So then we have a virus X that has been attenuated. If we took this and injected that in a human, it won't cause the disease that virus X causes, which we call virus eczema. And it will just make whoever got this shot just a little bit ill, but not sick. And in the process, the body will produce antibodies against virus X, right? So you get the attenuated version. The other option will be to inactivate virus X. So you totally kill virus X and then inject it as the vaccine. The body senses certain structures on this virus, notices it's not part of the human body, makes antibodies, and so you get your immunity to virus X. Okay. Now, what are some of the pros and cons of doing it this way? If you take the attenuated version, where you don't kill it all the way, well, one, the attenuation might not be enough, so that when you inject the virus into the human body, it actually causes disease, which is bad. Two, it's a long process. So when you need a vaccine quickly, like we do now, it might take a while to get that done, right? Now, if you take the inactive version, it's not as strong. So you might not get what you call sterile immunity, right? You might have to give several boosters, right? So there's always been the need to find another way of making vaccines which are more dependable and more effective. Enter mRNA. Okay, so how does this work? All right, let's take our virus X again. So this is the envelope. Okay, now under the microscope, we realize that virus X has these structures on it, actually. And of course, this is your genetic material. And then we realize that this and that these structures on the virus envelope, that's what actually pokes through the human cell and allows virus X to enter the human cell. And we also found out that when the body makes antibodies to virus X, it's actually making antibodies to these structures over here, right? We found this out. So we come to the next step. And the next step is, why don't we then just get this piece here or that piece here, inject it into the human body so that it causes antibodies instead of taking the whole of the virus X? So instead of, this is just part of virus X, and this is that structure on there that pokes through into the human cell. If we could capture this piece and inject it into the human body, then we don't need all of virus X with all the genetic material in it. Do I make sense till this point? So then how are we going to get just this piece? Now, remember, this is the genetic material, okay? RNA in this case. This genetic material codes for everything on virus X and in virus X. So that means part of this genetic material codes for this little structure here, this structure that we'll call, for lack of a better word, the umbrella. All right. So if we're able to sequence the genome of this virus X, so if we're able to sequence this, so CATGCTA, and so on. If we're able to sequence that, we should be able to tease out which portion of this whole genetic sequence makes for the umbrella, right? Then we could get nucleotides, arrange them, and make the genetic code that makes just this umbrella. And if we inject that code into a human body and the code gets into a human cell, it's going to make the umbrella. As soon as the cell in the body, sorry, presents this umbrella outside the cell. So let's say you have a human cell here now. It's injected with the umbrella code. And the very minute this gets in, the cell makes the umbrella. So all of a sudden, you have an umbrella sticking out of, say, a liver cell, for lack of a better example. The body is going to immediately know that this doesn't belong to it and it's going to make antibodies to it, right? So now you have antibodies to this. If virus X then attacks this person, there are already going to be antibodies in this person because of that injection. And the antibodies are going to inactivate virus X. And we say this person is immune to virus X and the disease it causes. So that's the whole theory behind mRNA vaccines. Okay. If we take SARS-CoV-2, that's what they've done. So you have with the spikes, they realize that this spike protein is very important in how the virus enters the cell. They also realize that it's a very, very stable part of this virus. The viruses have a habit of mutating. So they got the genetic material for just the spike protein, made mRNA out of that, and that is what now they are injecting into humans. And the human cells then produce the spike protein. And the minute the spike protein shows up, the body realizes this is not human and they make antibodies against it. So then that person becomes immune to SARS-CoV-2 and cannot develop COVID-19. I hope that makes sense till now. Now, the problem is that mRNA is basically protein and nucleic acid. And if you inject that raw bit of material into the human body, The enzymes in the blood are going to knock it out in no time. It's not going to get to the cell at all. So they have to find a way of getting this piece of RNA that they've made in the lab into the human body. And two ways have become very evident. Now, the human cell, as we know it, also has an envelope. So this is your cell. And this envelope is made up of mainly lipids. That means if you're able to create a shell of lipid and you put this piece of mRNA in there and you inject it, this is going to then attach itself to a cell and it's going to fuse with the cell, and the mRNA is going to wander into it. And so then it's going to get in here, and soon you have your spike protein sticking out. And that's the method Pfizer and BioNTech, as well as Moderna, are using. Big difference between the Pfizer and the Moderna one is the nature of the lipid particle that they enclose it in. The Pfizer one is a bit more unstable. The Moderna one is more stable. You need this to be at minus 94 degrees Fahrenheit, and you only need 4. So that's the big difference between the two. They seem to be both very efficacious. And though we didn't get it from the initial report from Pfizer, Moderna seems to also reduce the severity of a COVID infection. All right, next. What is another way of presenting this piece of RNA that codes for the spike protein to the human body? The guys at Oxford, working together with AstraZeneca, they use a very different method. They got a virus called the adenovirus. There's a type in humans and there's a type in chimpanzees, right? The adenovirus usually causes the common cold, among other things. Okay. So a lady there called Sarah Gilbert did a lot of the initial work. She got the adenovirus from the chimpanzee and basically gutted it out till it was just a shell. Whatever genetic material was left in there was basically useless. And then she took the mRNA for the spike protein from SARS-CoV-2 and stuck it in here. And initially, they took a line of human cells, I think lung cells, and they infected these human cells with this composite of the adenovirus gutted out with mRNA. And they noticed that the mRNA was able to get into these lung cells. So that is their composite, the CHAdOX, I think 1. That's the Oxford version, Oxford AstraZeneca. Okay. So far, we don't know how efficacious this is, but I bet it's probably as good. The only problem with the virus vector vaccines is that sometimes some human beings have antibodies against the adenoviruses, and that's why the Oxford group used the chimpanzee, because it's highly unlikely that any human has antibodies to a chimpanzee adenovirus, right? So that is the basis of our mRNA viruses, and it's really 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 amazing. Nobody believed it will work. Most people thought the efficacy will be 70% or even 50 to 70, and we are seeing as 90, 94%, which might give us sterile immunity, which means that it really makes you immune. Um, it kills off the virus totally, and prevents you from getting it altogether, right? And lastly, let's talk about efficacy and effectiveness. Okay. So the efficacy of the vaccine is what they measure when they do the studies in the controlled environment. And so now the efficacy we are finding somewhere between 90 and 95%. The effectiveness is when you really roll it out into the general population. Would it be 90 to 95%? It might not be. It might be lower for several reasons. One being that this is a two-shot vaccine, and somebody might get the first one and never get the second one, right? It happens. Um, Maybe production might affect it, maybe the storage might affect it, but there are sometimes differences in efficacy and effectiveness once you roll it out, and we're only going to find out. But the main thing is that we have two vaccine constructs that look very promising. And even though we won't get it tomorrow, the fact that it's around the corner and it would help us probably get this outbreak under control is extremely exciting. I hope this helps. And if you have any questions, go ahead and just shoot it out. Send me a message, post under this, whatever you want. I hope this helps. Thanks.